Good afternoon, everyone. Good to see you here. Thanks for coming. On behalf of the Center for Brain Health, welcome to Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Francesca Philby. I'm a professor in the School of Behavioral Brain Sciences at the University of Texas at Dallas, and a scientist here at the Center for Brain Health, where I direct the neuroimaging of reward dynamics, where we use addiction as a model for reward dysfunction. As a reminder, we will have a few minutes at the end of our speaker's presentation today to answer some questions. And so for those who are joining us virtually, please use the Q&A function. And for those here in person with us today, please have your questions ready to ask. We'll get to as many of your questions as we can at the end. As a reminder again, this presentation is being recorded. For those who don't know much about us, the Center for Brain Health is a cognitive neuroscience research at the University of Texas at Dallas. We have dedicated the past 3 decades to exploring neuroplasticity and the brain's amazing lifelong potential to get stronger and work better. Now, without further ado, I'm really excited to welcome our speaker today, Dr. Florence Chiang. Dr. Chiang is an assistant professor and an NIH First Scholar in the Department of Radiology, Advanced Imaging Research Center, and O'Donnell Brain Institute at the University of Texas Southwestern Medical Center. Dr. Chiang completed her medical graduate and residency training at the University Texas Health Science Center at San Antonio. She graduated from the School of Medicine with a distinction in research and was elected to the Alpha Honor Medical Society. She then completed an integrated diagnostic radiology residency PhD program by the American Board of Radiology Holman Research Pathway. So she did all of that all at once. Dr. Chiang earned her doctorate in radiological sciences from the Graduate School of Biomedical Sciences and received the Armand J. Guarino Award of Academic Excellence. She completed a 2-year neuroradiology fellowship at Massachusetts General Hospital, Harvard Medical School. In terms of research, Dr. Chiang's research focuses on developing and translating computational neuroimaging methods to identify and implement imaging biomarkers of disease, progression, and treatment response in multiple sclerosis and other neurodegenerative diseases. Her current funding is plentiful, but it includes the RSNA, have to go to the back of the page, Fellow Grant and Ralph Schwager Fellowship Award. Her prior grant support includes another RSNA grant, Research Resident Grant, Julio Palma's Award for Excellence in Radiology Research, and a couple of NIH awards, R-level, R25, and R03, which for most of us here in this room know how competitive those are to obtain. Dr. Chiang has also authored multiple publications and continuously presents her work nationally and internationally. So she is very high in demand. So I'm glad to have you here, Dr. Chiang. Thank you.
Hi everyone, I'm Florence Chiang. Thanks so much, Dr. Philby, for the introduction, and thanks to the organizers for the opportunity to speak. So I'm delighted to give this talk today on functional network modeling and microstructural imaging of neurodegeneration in multiple sclerosis as part of this lecture series. Today I'm going to show you some of the work we've done in computational neuroimaging in multiple sclerosis, where we've incorporated various imaging modalities, including coordinate-based meta-analysis, resting state fMRI, and high-gradient diffusion MRI. So we'll begin with a brief overview of MS and touch on the current imaging methods that are available. I'll explain why we need better imaging methods to monitor MS disease progression. Then I'll talk about a few computational neuroimaging approaches with application in MS. So we'll start by going over a biomarker development strategy using coordinate-based meta-analysis. This includes a discussion of the network degeneration hypothesis and extension in neurodegenerative diseases such as MS. And I'll also share with you our findings from testing the atrophy-based functional network model in resting state fMRI. Finally, I'll present our more recent findings in microstructural imaging of MS using an ultra-high gradient MRI system. I'll also highlight the potential for further assessing functional and molecular components of neurodegeneration in MS simultaneously via PET-MR. So multiple sclerosis is an immune-mediated disease of the central nervous system. So MS usually presents at a highly productive stage of life when people are planning families and building careers. So MS can have a significant impact on affected individuals, their families, and also society. So the growing arsenal of disease-modifying therapies really offers opportunities to reduce disability and also to extend survival of people with MS. But currently there's no cure, and the etiology of the disease remains incompletely understood. Here are a few highlights from this epidemiology survey. The estimated number of people with MS worldwide in 2020 has increased to 2.8 million, and this estimate is 30% higher than in 2013. And MS prevalence has increased in every world region since 2013, and it's estimated that someone in the world is diagnosed with MS every 5 minutes. MS is considered the leading cause of non-traumatic neurologic disability in young adults in many countries, and most of these patients are diagnosed in their 20s and 30s. There is also increased recognition of pediatric onset MS, with over 30,000 cases identified. Globally, the females are twice as likely to have MS as males, and the ratio can be as high as 4-to-1 in some countries. Now, to provide some historical backdrop, MS was first described in 1868 by Charcot, who was a French neurologist. He noticed that symptoms experienced by MS patients appear to be multiphasic, with initial difficulty in walking and maintaining balance, and then progression to tremors and ultimately severe physical disability. Charcot was a proponent of correlating clinical observations with anatomic and pathologic investigations. He first described MS pathologically when performing an autopsy on one of his servants who developed MS symptoms. Figure 4 here shows his drawing of the findings, where he noted plaques in the brain and spinal cord. We now know that these plaques occur due to damage to myelin, and myelin is found mostly in the white matter in the brain and spinal cord, and is important in insulating axons to provide efficient signal conduction. This tissue sample shows macrophages devouring the degradation products of myelin in the acute phase of disease. And this sample on screen right is showing plaques in the chronic phase with a relatively hypocellular region of myelin loss in the periventricular white matter. So because of these initial observations of lesions and plaques, the classical definition of MS is that it's a disease of white matter. Regarding the clinical manifestation of MS, symptoms can vary from person to person and may include disabling fatigue, mobility challenges, and also cognitive changes, as well as vision issues. And early diagnosis, accurate monitoring, and timely treatment are critical to minimize disability. This is the Expanded Disability Status Scale score, or EDSS scoring system, and this is used to assess the degree of disability of MS patients. And the EDSS scores range anywhere from 0 to 10, 10 being the most severe. Typically, patients start needing ambulatory assistance at EDSS of 4 to 6. In terms of the clinical course, there are two main subtypes of MS, the relapsing remitting and the progressive. So in relapsing remitting, there are periods of remission between the attacks, and in the progressive subtype, the disease kind of accumulates over time without any remission. But more recent evidence suggests that the disease manifestation is actually on the spectrum, with disease progression seen in all subtypes of MS. So here are a few images of conventional MRI methods, so standard of care clinical imaging of MS. These are quite sensitive in detecting demyelinating lesions on structural T2-weighted imaging. These lesions are commonly found in the periventricular white matter, but can also affect any other white matter brain region, and in the spinal cord as well. But due to the relatively heterogeneous nature of these demyelinating lesions, the association between clinical findings and radiological findings on conventional imaging is quite limited. And this has been termed the clinical radiological paradox. So perhaps there is a need to reframe the approach taken in imaging MS patients. So recently there's been more research on the significance of neurodegeneration and gray matter atrophy in MS. In this study, there's evidence that the gray matter fraction was better correlated with clinical features, including EDSS, compared to white matter fraction. And their results also indicated that the gray matter fraction explained more of the variability in clinical measures than white matter lesion load. Overall, the study concluded that in MS, gray matter atrophy reflects disability to a greater extent than white matter atrophy or lesions. And a more recent study also concluded that subcortical gray matter volume loss, in particular the thalamus, is a strong predictor of cognitive performance. In this longitudinal study published in Neurology, they looked at T1-weighted MRI of nearly 1,000 patients over a period of 1 to 2 years, and they found that the rate of atrophy appears independent of the MS subtype, and that this occurs throughout the clinical course, including early in disease. And beyond white matter changes, recent evidence suggests that neurodegeneration involving gray matter is also an important component throughout the disease course in all MS subtypes. Specifically, neurodegeneration in gray matter is localized, occurs very early in disease, and affects both cortical and subcortical brain regions. So this is a recent position paper by the North American Imaging and MS Cooperative. They argue that there's a need for finding better ways to characterize gray matter changes, to come up with methods that go beyond the identification of overt, irreversible structural volume loss. And these efforts have may lead to better understanding of the progression and discovery, the disease progression, and also discovery of medications that would halt neurodegeneration and promote repair. In 2016, a paper was published in Brain by Steinweg and colleagues. They observed that gray matter atrophy in MS is not global and not random, but occurs in a regionally specific pattern, affecting only certain gray matter regions. Since MS is a heterogeneous disease in general, the fact that there is a non-random pattern of gray matter atrophy is quite interesting. But now we have another problem. Why does this occur? So in a follow-up commentary by Chard and colleagues, several hypotheses were proposed. Of all of these, network mediation seems most plausible and relatively straightforward to test. So network mediation is a relatively new concept, at least in MS, but it has been well described in other neurodegenerative conditions. Celia and colleagues reported network-mediated patterns of gray matter atrophy in several dementia syndromes. They first described a pattern of localized gray matter atrophy in dementia patients. Then they examined these atrophy-prone regions in healthy controls and found that these brain regions are functionally connected in healthy individuals. So the conclusion was that brain regions that co-atrophy also seem to co-activate and are functionally connected. And this describes the network degeneration hypothesis, which states that atrophy patterns correspond with healthy functional network architecture. So in this figure here, there are distinct atrophy patterns in different types of dementia, which demonstrate a convergent pattern of functional connectivity in resting state fMRI of healthy controls. So we asked the question, is localized gray matter atrophy in MS mediated by functional networks? Now I'll talk a little bit about how we can test and also validate the hypothesis using a data-driven approach. In general, acquired data is hard to come by, and small sample sizes is a common issue, which really limits generalizability. So we can perform coordinate-based meta-analysis to come up with a model representing effects that are most commonly reported in the literature. We can use coordinate-based meta-analysis, which are reported typically in standardized XYZ coordinates, and these types of data are helpful when comparing results across studies. In 2013, a convergent pattern of gray matter atrophy in MS was identified meta-analytically, and this pattern correlated strongly with physical disability in MS patients. We wanted to confirm this atrophy pattern and extend these findings to test for interregional functional connectivity. This can be done using the BrainMap database, which contains coordinate-based data of both healthy controls and also various diseases, including MS. This is the BrainMap pipeline, which starts with the input of coordinate-based results from published literature. The database contains both structural and functional results. And the structural are typically T1-weighted MRI, and functional results come from task-based fMRI and PET imaging. And we can search and retrieve relevant data. Finally, we can use GingerRail, this application, to analyze coordinate-based data and determine whether there are consistent structural and functional results in the literature. And by using this pipeline, we identified 8 atrophy clusters, and by taking the AL peak values within these clusters, 17 Cs were identified. The AL map here shows regions of atrophy that were found to be most consistently reported in the literature. These include both subcortical, so deep gray matter, as well as cortical regions, and the effects load most heavily on the thalamus. And here's a side-by-side comparison of our atrophy pattern results with those that were published previously. Even though the methods were different, the results are comparable. And by using these atrophy regions, we identified interregional functional covariation using healthy control functional data from the BrainMap database. Here's the final node and edge model we derived in MS. The nodes represent atrophy-prone gray matter, which are specific in MS. And the edge weights demonstrate the presence of healthy functional connectivity between these brain regions. So overall, this node and edge model represents a functional network signature in MS. And we can apply the AFM model in resting state fMRI as a candidate imaging biomarker. Here's the overall workflow of applying the AFM model in resting state fMRI. First, ROIs were defined based on the nodes in the AFM model. Then the ROIs were used to sample the pre-processed fMRI time series data. And since each subject had 700 volumes of resting state fMRI data, there were 700 samples per node per subject. And each of these sampled time series provides data for observed variables and path analysis in structural equation modeling. So this figure just shows the AFM model applied as a path diagram in SEM. The unidirectional arrows for each path indicates a conditional probability, which represents functional connectivity. So these results were published in Radiology and demonstrated that the AFM model can serve as an MS-specific functional baseline. In this study, we applied the meta-analytically constructed AFM model in prospectively acquired data of 20 MS patients. The RMSEA was correlated with EDSS, and we can see that there is disruption of network integrity and also deviation from a healthy functional baseline as disease severity worsens. On the other hand, normalized white matter lesion volumes demonstrated a negligible clinical association. Next, we examined path level differences between MS and healthy controls, and we assessed the diagnostic accuracy of paths that were significantly different between MS and healthy controls. Our results demonstrated diagnostic accuracy of nearly 80% by using the AFM predictors in resting state. And in a validation MS data set, which was matched to the original cohort, we also tested the AFM model to see if the results can be replicated. Here we can see that the correlation of RMSEA with EDSS in blue demonstrated a positive association as before. So a couple of limitations in this preliminary analysis, you might notice that the EDSS range is more restricted in the replication data. Also, the replication data had only 300 volumes versus 700 in the original data, and since RMSEA is essentially a measure of error, it's expected that the absolute value is greater when there's less data. So here we can see that the data points are all shifted up on the y-axis, so vertically. However, the overall trend is very much similar to the original model. And repeatability of AFM metrics was also assessed using Bland-Altman plots. Plots A and B show results for 5 and 15-minute scans. We can see that the reliability is quite high. In both cases, achieved 95% limits of agreement and an acceptable interclass correlation coefficient of greater than 0.7. So our findings are in line with evidence in current literature. There was a recent review paper published in Neuroimaging Clinical talking about network collapse in multiple sclerosis. So the authors in this paper laid out several key observations. First, lesional pathology disconnects structural pathways, and this is perhaps conceptually the most straightforward. On the other hand, you have complex functional network changes that follow the clinical status of the patients. But there's a need to really better understand this structure-function relationship. Another concept is the idea of network hubs, which are brain regions most connected with other brain regions. And the evidence suggests that this network collapse occurs after the disease burden exceeds a certain network efficiency threshold. So once the network is disrupted, this may result in faster neurodegeneration and accelerated disease progression. So here's a recent review of fMRI studies in MS. Table 3 lists the most important resting state fMRI studies analyzing brain functional connectivity in people with MS in relation to clinical disability. About 25 papers were highlighted, including our radiology paper. So all of these studies essentially demonstrate evidence of network alteration with disease progression. Okay, so now shifting gears a little bit, I'm going to talk a little bit about our more recent work in microstructural imaging of MS. We can use diffusion MRI as an indirect probe of structures on the micron level, so such as cells and axons in the brain. And this is done by using the Brownian motion of water to infer the size and shape of all of these structures, which are on the order of microns. And as such, diffusion MRI holds great promise as a non-invasive imaging method for probing cellular structures in the living human brain. And we can use diffusion MRI to perform microstructural imaging. So let's talk a little bit about the hardware technology that's needed to perform microstructural imaging of brain tissue. So previously I worked on the Siemens Connectome 2 scanner at the Martino Center for Biomedical Imaging in Charlestown, Massachusetts. This is an ultra-high gradient system that was designed to be the ultimate diffusion machine, mostly focusing on tractography and mapping all of the major white matter pathways in the human brain. And using scanners like this, we can perform diffusion MRI at shorter effective diffusion times to probe micron-sized structures. So why do we need strong gradients for diffusion MRI? On a conventional MRI system, in order to get sufficient diffusion weighting, we typically play out the diffusion encoding gradients over a long period of time with a very limited Gmax, so the maximum gradient strength. And this in turn pushes out the actual measurements to very long echo times, which results in a decrease in signal to noise ratio. So the quality of the images are not as good, right? And with stronger gradient amplitudes, we can achieve a given diffusion encoding gradient area in much less time. So this allows us to essentially boost the signal-to-noise ratio by pulling in that echo time. And another benefit of having large gradients is that it enables stronger diffusion encoding, and we need these larger diffusion encoding areas to probe very small structures. And stronger gradients overall boosts this, what we call diffusion resolution. And high gradient systems can serve as an in vivo microscope. You can think of it as that, which allows us to interrogate tissue microstructure with micron level resolution. Here are simulation results showing the resolution limit of the Connectome 2 compared to Connectome 1, which was its predecessor, and to commercially available clinical scanners. So the shorter effective TE achievable with Connectome 2 doubles the SNR, which sets the resolution limit, which is denoted by these horizontal bars that you see on the graph, at a minimum axonal size of 1.4 to 1.6 microns versus on Connectome 1, which was 2.5 to around 3 microns. And the threshold for the resolution limit of axonal diameter estimation on Connectome 2 gets us much closer to histological measurements, so tissue samples, which is around 1 micron. And here we can see that at higher B values, there is increased SNR. And when we compare Connectome 1 to 2, there's better SNR at higher B values with the increased gradient strength. So in this particular example, with the higher SNR achievable at higher B values, we can push the resolution limit of axonal diameter estimation. So there are various, so that's the hardware portion. So now we can talk a little bit about the modeling approaches and the software, right? So there are various biophysical modeling methods using diffusion MRI. And these biophysical modeling methods allow us to assess different tissue compartments in vivo. And this is SANDI, which stands for Soma and Neurite Density Imaging. What's novel about this technique is that it allows us to probe multiple tissue compartments, including in gray matter. So here's our schematics of the current standard model of brain microstructure compared to SANDI. So in figure A at the top, that shows the current model, which treats tissue components in the MR voxel. So each 3D pixel or voxel in the brain image can be subdivided into two different non-exchanging compartments. And this is the intraneurite and the extraneurite space, so inside the tissue and outside of the tissue. And the total MRI signal is then given by the weighted sum of the signals from water molecules diffusing in each of the compartments. And figure B at the bottom shows the SANDI model, which subdivides the MRI voxel in the intracellular and extracellular non-exchanging compartments, but then it does one more step. Within the intracellular compartment, that's further subdivided into another two different compartments, so the intraneurite and the intrasoma fractions. So what this allows us to do is to look at gray matter in addition to white matter. So overall, this SANDI model allows us to look at both tissue compartments, white matter and gray matter. And here are tissue contrasts of the neurite and soma fractions of the human brain. On screen left, outlined by the blue box, we have a figure showing the comparisons between the intrasoma signal fraction compared to the histological images stained for brain cytoarchitecture. On screen right, we have a figure showing the comparisons between the intraneurite signal fraction compared to the histological images stained for brain myeloarchitecture. So these contrasts in the MRI maps show a remarkable similarity to the contrast from histological staining. And the quantitative intrasoma signal fraction provides a contrast related to soma density, which is most useful for assessing gray matter. And on the other hand, the intraneurite signal fraction provides a contrast related to neurite density, which is highly related to myelin in the white matter. And we've applied these novel techniques in multiple sclerosis, and the most recent were recently published in Brain Communications. In this paper, we demonstrated that MS features complex microstructural changes in normal appearing gray matter that's associated with neurodegeneration. And using a high gradient diffusion MRI technique, both hardware and software-wise, we can detect subtle microstructural changes, which has the potential for more sensitive monitoring of disease progression. And this is important because we can catch these changes likely before the overt irreversible changes that we see in brain atrophy. So in this paper, we show that there is a significant difference in soma density between MS and healthy controls. And then we also observed that the cortical cell body density correlates with thalamic volume. And the conclusion was that cell body density may be sensitive to the substrate of neurodegeneration and reflect disability status of people with MS. We took this a step further to evaluate the intrinsic and extrinsic microstructural factors that are contributing to atrophy within these individual thalamic subregions in MS. And we applied SANDI in using high gradient diffusion, MR, in both MS and healthy controls to assess these differences. So after parcellating the thalamus into 25 subregions, we evaluated the volume and also the diffusion metrics of each subregion. And the diffusion estimates included the intrasoma fraction, also the intraneuri fraction, so that's gray matter and the white matter, as well as the extracellular signal fraction, which is essentially CSF. And we compared the thalamic nuclei volumes and microstructural metrics between MS and healthy controls. We found that in the bilateral medial pulvinar subregion and in the right medial dorsomedial nucleus subregion, there is lower soma signal fraction and then higher extracellular signal fraction. So these nuclei also demonstrated lower normalized volume. And our results show that the medial and posterior thalamic nuclei are preferentially affected by atrophy. And the lower thalamic nuclei volumes are associated with microstructural changes, including decreased intrasoma signal fraction and decreased intrasoma signal fraction and also increased extracellular signal fraction in MS compared to healthies. So since these thalamic nuclei are bordered anatomically by the CSF in the third ventricle, our results suggest that there may be central spinal fluid mediated thalamic damage, which is driving thalamic atrophy in MS. We also wanted to assess network behavior of microstructural alterations in atrophy-prone gray matter. So we applied the atrophy-based functional network model, or AFM model, in high-gradient diffusion MRI using SANDI. The ROIs were defined at nodes in the AFM model, which were used to sample the soma density map of each participant. And we observed a lower soma density across all of these nodal regions for MS compared to healthy controls. And the aggregate nodal intrasoma signal fraction demonstrated an association with EDSS scores. So this is the disability status scale score that I described before. And as the EDSS score gets worse, the soma density also decreases. Then we observed covariation of cell body density involving these atrophy-prone gray matter regions, both in the subcortical as well as cortical regions. And this is shown here in this heat map. Our results suggest that the neuronal loss may be related in part to network-based effects. Okay, so during my talk today, we started with an overview of MS pathophysiology and touched on kind of conventional imaging used for clinical assessment. And we talked about the clinical radiological paradox and how there are limitations of using T2-weighted structural imaging to assess lesions for monitoring MS. We also discussed that neurodegeneration is a key component of clinical disability. Then I introduced the network degeneration hypothesis to you and showed how we tested this in MS by using the atrophy-based functional network model, or AFN model. I shared our results using a normative modeling approach to identify monitoring biomarkers in MS. Next, I shared our more recent work using microstructural imaging of MS, which utilizes multi-shell high-gradient diffusion MRI. We reviewed the benefits of using ultra-high-gradient diffusion imaging to improve axonal diameter, as well as cell body density measurements. And we also talked about SANDI, biophysical modeling approaches for assessing gray matter microstructure in MS. So in terms of future directions, our group is interested in development of multi-modal computational imaging biomarkers in MS. This will not only help to track disease progression and the disease burden in the clinical setting, but also has the potential to really monitor response to drug therapies. Additionally, we are implementing a novel PET radiotracer for microglial imaging in MS to further assess the pathologic mechanisms involved in disease progression. This will be done via simultaneous imaging acquisition of PET and MRI with a next generation PET MRI scanner. So the potential yield from all of these future works is a computational imaging algorithm for quantitative assessment of MS. In terms of clinical impact, the results could provide more accurate imaging monitoring of patients and also better prognostication. This would improve patient management in the clinical setting and would also impact the success of neurotherapeutic development in terms of non-invasively tracking more subtle neurodegenerative changes. And this is all not currently possible with standard of care imaging. With that, here is the team who made all of this work possible. We have folks in various disciplines, ranging from biomedical engineering, medical physics, neuroscience, and medicine. Our funding sources include the NIH and the Radiological Society of North America. Well, if you find this work interesting, please feel free to reach out anytime. I'd love to chat more. And if you're looking for a position, please also contact me as we are actively hiring. That's my email, and I'm also on X and LinkedIn. Thanks very much for your attention. Happy to take any questions.
Thanks, Dr. Chiang. We can take questions. There's one in the back there.
Hi, great talk, thank you. Two questions. First of all, you may have mentioned this. Were you looking at white matter hyperintensities or hypointensities when you were looking at white matter lesions?
So if you're referring to the kind of relatively minor component of the radiology paper, we were mostly looking at, usually people look at the hyperintensities on T2 imaging. Although on T1, it would look dark, right? It would be hypo on T1. Right. But that's the conventional imaging approach, is to use kind of volumetrics to assess T2 hyperintensities. But as you can see, and what I've shown, is that it really has limited associations with the clinical status of the patient, right? And so it doesn't really correlate very well, and that's a major limitation of current techniques.
Yeah, and so like I'm looking into white matter hyperintensities myself, and I'm just interested if you had any thoughts as to why you didn't see as much of an effect compared to gray matter atrophy on physical disability. And then also, I'm not sure if you do cognitive testing as well, but do you think that would show up as in the white matter hyperintensities having less of an effect? Would you see that in cognition as well? What do you expect?
That's a great question. So first, in terms of the white matter lesions, I think that, they're real. They're obviously there. But I think the issue is with current technology in terms of imaging. So I think that's like the tip of the iceberg. We're seeing the lesions, but there's more to the lesions, right? First of all, the lesions can be in MS, but it can also be in a lot of other disease, or even just normal aging. Like every decade, you get one of these. So, it's kind of like a normal person, healthy person, right, without MS. And so that's a major limitation that's not really specific. Another thing that a lot of people are working on in terms of lesional assessment is to further characterize these lesions. So it's not just the volume of these T2 hyperintensities. It's also in terms of, have you heard of like central vein sign, right? That's another thing. We can use susceptibility weighted imaging or any sort of like T2 star weighted imaging to assess these central vein signs, right? Another thing is the paramagnetic rim. So we can also use susceptibility weighted imaging to assess whether there's iron deposition surrounding these lesions, and that might clue you in on perhaps chronic inflammation, ongoing inflammation in these lesions. And so when you take kind of this additional step in terms of characterizing these lesions, you'll see that perhaps not all of the lesions will exhibit these signs. And so first, it may be not specific, but another thing, it could be just like burnt out lesions. They're no longer active. And so I think there are a number of reasons why the lesions just on T2, like if you're just saying counting the number of T2 hyperintensities, why that doesn't really correlate with clinical disability, right? So, yeah.
Can I just add, Darren also doing the texture and shape of the lesions. So those are some other lesion characteristics that I just wanted to throw that in.
I think that's very important, and yeah, thank you, Dr. Ripma, for bringing that up. Definitely having that additional layer of characterizing these lesions would be helpful. Yeah.
So I have another question since I have the microphone.
Please.
And that is, so in the AFN model, I noticed that the nodes are populated mainly by subcortical structures and motor. Is that my
sensory
right?
motor and
right.
Some gyro regions.
Sensory motor, yeah. And so I was wondering, in terms of the clinical radiologic paradox, how you think that model informs, as the previous questioner was asking, questions about the relationships of these changes to cognitive changes.
Yeah, I think that's a great question. I think the lesions are certainly a component of cognitive changes because, of course, they're mostly involving like demyelinating lesions, right, the white matter. And so that really does affect signal conduction between different brain regions, so that's definitely important. But I think there may be either downstream or altogether separate independent component of disease progression, and that's why there's so much work going on in terms of trying to identify these neurodegenerative processes, and cognition is huge. I mean, that's definitely a very important endpoint. And in current clinical trials, I kind of alluded to this, but to talk a little bit more about that, currently they're using essentially these whole brain volumes, right? And so first, these could take years. Usually the studies are like 5 to 10 years. But that gets really expensive for clinical trials. You're thinking, I want to wrap this up in a couple years, right? And so first, it takes a long time. And second, I think more importantly, is that these overt brain changes are really irreversible at this point. And so I think the benefit of using advanced imaging is to try to identify changes that come before that, the more subtle changes. And perhaps these would show a better correlation in association with cognition, because obviously if there's atrophy overtly by eye, like I can see it, then definitely there will probably be some cognitive changes, right? But I think the more subtle changes, usually those kind of patient populations would definitely benefit from some sort of quantification of their disease, which is real.
Yes, I have a question and I'd like to make a statement. In regards to the cerebrospinal fluid involvement in affecting the thalamus, has any studies been done in looking at this fluid and what may be the etiology, what may be the components that's affecting the thalamus? Has any work been done in that area?
Yeah, so I think there's ongoing work with that. Usually not in imaging. Usually we're thinking more basic sciences, right? Because it could be due to a number of things, like it could be toxic as well from the CSF itself, but also possibly a mechanical etiology as well, given the location. So I think that's work in progress in the field.
The statement that I'd like to make is I really appreciate this lecture and seeing your presentation. My training was in the 70s in psychiatry, dual residential psychiatry and neurology. And in the 70s, mid and late 70s, we didn't even have a use of a CAT scan or MRI. And looking at diseases like multiple sclerosis. So this is really a delight to see the progress of the sciences, and to see this from the 70s up to 2025 is really impressive to me.
Thank you very much. Appreciate that.
Avery.
First off, thank you for such an interesting and engaging presentation. My questions are going to be a lot less technical, but the only question I really have is I noticed that the prevalence of MS seems to be drastically increasing. I think the number was 30%. And I kind of wanted to ask if you guys had any hypotheses for that.
Yeah, I think because the etiology of MS is still unclear, like people have been looking at, is it vitamin D levels, is it a viral etiology? To be honest, like it's hard to say. And I think that part of it may also be that there's increased awareness of MS and how it manifests, right? The cognitive portion, I think, is so important because a lot of times these will be misdiagnosed as something else. That could be, say, psychiatric, for example. And so I think now we're seeing, there's serum markers, there's CSF markers, there's quantitative imaging evidence showing that there's something wrong, like at the pathophysiological, so that at that level. And so I think it may be in part due to just increased awareness of this entity. Yeah.
Thank you for a very interesting presentation. What is the availability of the hardware and the software within the general patient community?
Yeah, thanks very much for that question. So the Connectome scanner that I mentioned is the only one in the world. It's a research-only scanner, limited, definitely in terms of in the community, right? But there is actually recently, last year, there's an FDA-approved scanner now, and we'll be getting probably one of the first ones in the U.S. at UT Southwestern, and so it's called the SEMA X. It's by the same vendor, Siemens, and so it has slightly less powerful gradients, but usually on those powerful scanners, sometimes you don't hit all the way to 500 anyway, and so I think it would still be much better than what we have in terms of conventional imaging, like the MRI scanners in the hospital. And so we'll be getting two actually at UT Southwestern, and I think that would be very beneficial in terms of having that availability for the patient population. One will be on the research side, but the other one will be in a hospital setting. And so I think slowly, as people start doing translational work like this and method development, there will be a shift in terms of the technology, hardware and software-wise. And software, of course, is always about standardization, whether it can get FDA approved. And so that's kind of part of my work as well. We do have a platform that eventually we would like to have it disseminated, in this clinical setting. And so it would essentially be like an automated pipeline that images would go through, and then I'm a radiologist, so it would go on a PACS system for the radiologist to incorporate as part of their report. But that would provide that sort of quantitative component. It's kind of, if you think about it, kind of like a blood marker, if people test for their blood sugar, right? So this would be another set of numbers that the clinician, the neurologist, can take into consideration.
Dr. Chiang, really nice work, very
sophisticated. My question is, if you were to guess from treatment effectiveness, because many people that do have MS, there's treatments, what do you feel like your measures, which one do you think might change and how if the treatment was working? You said it could be a marker of treatment effectiveness. What do you think might change?
Yeah.
Is there something, and what are you most hopeful for or in keeping up with some of the treatments for MS? And is it a heterogeneous disease that there probably will be, like Alzheimer's, many causes that bring out MS?
Yeah, thanks, Dr. Chapman. So I think in terms of the use of these biomarkers in clinical trials, I think that's what you're referring to, like as an endpoint, an exploratory endpoint, or primary endpoint. Ultimately, I think that it would likely be the resting state fMRI measures that I'm describing. I think all the other measures, such as the microstructural imaging and even PET, take a little bit longer. I think just feasibility, right, in terms of the table time in the clinical setting. And so these are kind of validation efforts, I think, that are very important. But in terms of feasibility, it's probably going to be the resting state fMRI because that can be implemented on a lot of scanners. And so whenever we run clinical trials, that's going to be multi-site. We have to consider the different settings. And so I think for resting state, that would probably be the most useful tool. But also from a physiologic and biologic standpoint, I think the functional changes would likely pick up these effects earlier. And so I think that has the most potential in terms of closely monitoring the disease progression, right? So we don't want to lag too far behind. Yeah, one word about the PET imaging, of course, that's a little bit more invasive, right? So, and there's some radiation associated with that. And so MS patients typically get scanned every 6 months, and so it's pretty routine. And so we need to find something that they can tolerate every 6 months. And so that's another consideration.
Yep. Great talk, Dr. Chen. Thanks so much. I just have a question. Because you mentioned like MS is a heterogeneous disease, and I expect there will be like individual differences in here. And do you think this functional network mediated pattern can be like more personalized, or the network pattern can be dynamically changed during the disease progressions? Because you also mentioned you're gonna focus on the computational biomarkers in the future based on the pattern. So I'm just wondering, what's your comment about that?
Yeah. So these studies so far are, at least in our group, is cross-sectional. So, but we do anticipate doing a longitudinal study in the near future, and I would expect actually changes that we don't know about. Again, that time lag I think is important to figure out in terms of the changes clinically and the imaging findings. But I do expect a non-linear trajectory potentially across time, and so I think that would warrant a larger study at that point. But yeah, great observation.
I will ask a question and then we'll go to our virtual question if you allow me. I wanted to follow up on your response to Sandy's question about using resting state or expecting resting state fMRI to be to change along with improvements of in treatment, in response to treatment. I noticed in one of your slides on the resting state slide, the last one before you started the microstructural section of your presentation, that the directionality of those resting state results were mixed. And so I wonder how that would, like what your thoughts are in terms of the expected changes of resting state with as a marker for response to treatment when it doesn't seem to have a clear kind of pattern.
Are you referring to the directionality of the paths?
Or the directionality of dysfunction, where you have increased resting state connectivity for a group of individuals, and then I saw on another line that there was decreased. So the directionality of change seems to differ from study to study.
Oh, I see. I think you're referring to the radiology paper, our results. So actually the first graph was in terms of the model fit. It's really a model fit measure, so how disrupted is this network? So that was the line that basically correlated strongly with disease progression. But actually the bottom graph, that goes the other way, was actually referring to lesions. So I think I didn't highlight that enough, but that was actually referring to lesions and the fact that it really doesn't correlate very well because it's a structural measure and also pretty limited in terms of assessing the lesions. But in terms of the functional connectivity, there may be, there are multiple paths in this AFM model, and some of which may be increased in terms of functional connectivity, others may be decreased. These are all essentially correlation coefficients, right? But we're actually looking at the model at a global level, so not individual edges. We're looking at it from the whole model level because, as you mentioned, and a few of you remarked, that throughout the disease there may be changes over time, right? It's dynamic brain function. So at this point, we're really interested in the entire network, how it deviates from a healthy functional baseline. And so while one edge or one functional connection may be increased, the other may be decreased, but as a whole, how is this deviating from the model that we've come up with? And so I think that's in the near future, we have planned research to kind of probe a little bit deeper in terms of each edge and also how that relates to cognition, right? Different domains. But at this point, our data is really just describing at a global level that we do see a very clear pattern of functional network disintegration and disruption for the patients. But I think that those would be helpful markers as well at the sub-model level.
Thank you. We're going to go ahead and take a question from our virtual audience. Here's one that asks, if you see the diffusion MRI and the AFN model as a potential assessment tool for Parkinson's or Alzheimer's or other neurodegenerative disease besides multiple sclerosis?
Yeah, that's a great question. I think that in neurodegenerative diseases are very interesting to me. I think that taking a more functional network approach and assessing these brain regions from this network-based approach has really great potential in terms of identifying these changes that really go beyond just volumes, right? That there's decrease in volume of these particular brain regions. And we actually, in my prior lab, in Peter Fox's group in San Antonio, we do have a model for Alzheimer's disease as well. And so that's currently being implemented in resting state as well. Yeah, so we have different disease-specific signatures, essentially, using functional connectivity.
Excellent. If there aren't any more questions from the audience, I'd like to echo the gentleman's statement that it's amazing how we've progressed through technology, but I think even more exciting are the kind of people, the pioneers who take advantage of those tools. So thanks, Dr. Chiang.
Thanks so much for having me.